Alright, so this is a quick introduction to Mendel and alleles. Mendel was a monk and teacher. He and his fellow monks were fascinated with the breeding of plants. Around 1857, he started breeding garden peas in his monastery's garden to study inheritance. He began experimenting with plants that were true breeding. And this means that a true breeding plant crossed with another true breeding plant, all the generations will be the same characteristic. So a blue-petaled plant crossed with another blue-petaled plant would give all blue-petaled plants. And there could be a true breeding white-petaled plant, white crossed with white, and all their generations down the line crossed with each other are going to be white-petaled. So the cross of two true breeding species is called hybridization. So in our example, we could have a blue-petaled plant crossing with a white-petaled plant. And when crossing two initial species, this is what's called the P generation. And any children of the species, whatever they may be, is called the F1 generation. And if you take an F1 generation species and cross it with another, this is what gives you your F2 generation. So Mendel experimented with traits that had two different forms. One was dominant and one was recessive. Today, when we write the gene symbols, we use capital letters to denote dominant traits and lowercase letters to indicate recessive traits. Homozygous means that both letters are the same. Both alleles are the same, while heterozygous means they're different in the same individual. So, as an example of this, if blue was dominant, homozygous dominant, for petal color, it would be BB. And if white was homozygous recessive, it'd be BB lowercase. And a potential cross of these two could give us an F1 generation that has the following characteristics. And this is a Punnett Square, and this is how you determine what an F1 generation would look like in this example. So BB for this individual, and you use lowercase b, and then you just cross it. BB, BB. And in this example, the blue plant is homozygous dominant for petal color, while the white plant is homozygous recessive in both positions. So we're going to get all the same individuals with all the same characteristics. So in the F1 generation, we're going to have all blue and no white. If we take this a step further and we go into F2 generation and we cross an F1 and an F1, we'll see the following. We take BB, since we know that this is BB, and this is BB, and this one and this one. So if we cross these two, we'll get the following. So we have one that's dominant homozygous, two that are dominant but heterozygous, and one that's homozygous recessive. So in this example, we see 1, 2, 3 that are blue, and 1 white. And 1 white. So in this example, the ratios of what we see for F1 is 100% blue, or 1 over 1. And in F2, we see 75%, or we should write 3/4 blue, 1/4 white. And that's the ratio for an F2 generation. So now I'm going to move on to an even more challenging problem, and we'll see if we can solve it.